like to begin by thanking the organizers and sponsors of this terrific meeting for the opportunity to introduce Iconic Therapeutics for you today. We're pursuing breakthrough opportunities in retinal disease and cancer. The thread that links these two for us is tissue factor, a protein and pathway important in the pathophysiology of several important diseases. We're targeting this currently undrugged pathway by leveraging advances in tissue factor biology and new scientific understandings to address significant therapeutic needs. For example, in wet AMD, the critical need for disease-modifying agents and new mechanisms of action. And in cancer, we have the opportunity to treat the tumor, its pathologic vasculature, and the microenvironment with a single targeted agent. Our lead clinical candidate, ICON-1, is in phase 2 for wet AMD now, and we're expanding our portfolio of development programs. Next up for us, we plan to file an IND in 2016 for ocular melanoma. We've been fortunate to attract a world-class management team, very experienced advisors, and a terrific board. We benefit from a strong financial position and a suite of intellectual property, both in-licensed from Yale and developed in-house at the company. In addition to wet AMD, we believe there's great opportunity for the tissue factor pathway and other retinal indications, in particular proliferative diabetic retinopathy and diabetic macular edema. And in addition to ocular melanoma, we're pursuing several lines of research in solid tumors and developing second-generation molecules with scaffolds and properties tuned specifically for systemic delivery for these diseases. Tissue factor is a protein and a pathway that's been well studied in hematology and oncology. It plays a key role in the normal regulation of coagulation and innate immunity, but in disease, overexpression drives pathologic angiogenesis and inflammation, increases metastatic potential, and promotes escape for immune surveillance, which has significant implications for immuno-oncology agents. Our lead clinical candidate, ICON-1, is a first-in-class human immunoconjugate fusion protein, which has been specifically engineered to safely bind tissue factor and attack tissue factor overexpressing cells. Now, normal retina does not express tissue factor. However, in response to chronic hypoxia and inflammation led by inflammatory macrophages and inflammatory cytokines, tissue factor can be directly expressed on CNV and secreted into the vitreous. Once tissue factor levels are increased, it drives increased VEGF release directly via a positive feedback loop, and also drives the amplification of angiogenic cytokines and the release of several pro-inflammatory cytokines, including IL-6 and IL-8. So together, increased levels of tissue factor drive both angiogenesis and inflammation, the pathologic hallmarks of wet AMD. We've studied ICON-1 in several preclinical models of wet AMD and observed both prevention and regression of CNV production after a single dose, and in larger animal models, a nice dose response and an excellent safety profile, in particular without any evidence of coagulopathy or bleeding abnormalities. On the basis of that work, we filed an IND and completed our first-in-human phase 1 study in the United States. This single-dose study was, of course, focused primarily on safety with broad inclusion criteria. Half of the subjects were newly diagnosed and half heavily pretreated. Safety outcomes were good with no ocular or systemic safety issues and no immunogenicity seen in this single-dose open-label study. There were signals of potential dose-related biologic activity, with individual patients experiencing visual acuity increases, reduced retina fluid, and reduced leakage on angiograms. The graph on this slide illustrates the clinical journey of one such patient who was treatment naive at entry in the 0.3 milligram dose group. This slide shows imaging from this patient. At screening, OCT demonstrated significant intraretinal and subretinal fluid, and on angiography there was an actively leaking classic CNV lesion. 5 weeks after receiving a single dose of ICON-1 without any concomitant anti-VEGF or other therapy for this newly diagnosed AMD patient, the OCT demonstrates significant reduction in the intraretinal fluid, and the angiogram is markedly altered. We're currently enrolling the eMERGE study. eMERGE is a phase 2A, double-masked, active-controlled, multicenter trial in the United States. we're enrolling 90 treatment-naive, primary active CNV patients to 3 dose groups: ICON-1 alone as monotherapy, ICON-1 in combination with Lucentis, and Lucentis at standard doses as an active control. The study design includes 3 months of monthly therapy as induction, followed by 3 months of follow-up with protocol-driven criteria for retreatment. The trial is enrolling now, and we expect it to read out in the latter half of 2016. As I mentioned, tissue factor is broadly applicable in cancer, where it's a marker for aggressive tumor growth, poor prognosis, and is central to the process of metastasis. Of particular interest to us here today, tissue factor is overexpressed not only in cutaneous melanoma, but also in ocular melanoma. We have positive data in xenograft models of melanoma and are moving to file our IND for primary treatment of ocular melanoma next year. We've assembled a leadership team comprised of individuals deeply experienced in the global development of retinal drugs, as well as in development of biologics for cancer. We're grateful to our current lead investors, MPM Capital, HIG BioVentures, and Lundbeck Fund Ventures. We're very proud of our scientific and clinical advisory board shown here, and of our passionate and committed investigators, several of whom are here today. In summary, Iconic Therapeutics is pursuing breakthrough opportunities in both retinal disease and cancer. Led by deep knowledge of tissue factor biology, we're developing unique therapeutic applications based on known and emerging science to address critical therapeutic needs in important diseases. We're developing a multiple product platform, and as we look ahead to 2016, we'll be excited to share several clinical milestones with you. Thank you.